Hello, lovers of life, and welcome to another exciting episode of General Biology. Today's topic is the structure of DNA. The key points that we're going to look at today is we're actually going to learn what DNA is, we're going to figure out why it's important, we're going to find out where we find it, what it's made of, and then how those pieces are put together to make the structure. Okay? So there's a representation of it. And doesn't it kind of look like a ladder twisted? Well, remember that because I'll reference that a lot. So what is DNA? Well, DNA is a macromolecule. Remember, that's a really big molecule, and it's known as a nucleic acid. That's just a type of macromolecule, like protein or carbohydrate or lipid. Its full name is deoxyribose, which is a sugar, deoxyribose nucleic acid. And if you look at this picture, each one of those little bubbles on there is really an atom. And so that's a whole bunch of atoms put together. So let's talk about genes, because that's what DNA holds, but not those kind of genes. DNA actually stores our genetic genes. It's spelled different. And genes are just little sections of DNA. And when all those DNA sections join together, they form a full strand of DNA. So see, that blue is the whole strand of DNA, and the pink is just that one part of the gene. So that's really, I guess, how to think about it is the gene is what's important, and I'll tell you why in just a second. But when you put all those genes together, you got your full strand of DNA. Okay, so what does the gene do? Well, the gene stores the instructions for building a protein. Okay, now proteins, why are they important? Proteins make a lot of the structures in our body, and we've talked about some of those. I'm going to show you like the cell membrane and so we've got the protein gates, protein channels. Proteins are used for all sorts of things in our body, but they're also the workers. And that's really where I think they're the most important almost. So like this motor protein that you guys have seen, that's a protein. And then we call them enzymes, okay? So if we're going to sum up why DNA is important, this is what I would say. It holds the information that makes all the proteins, that makes many of the structures in our cells, and does most of the work in our cells. So what cells have DNA? Well, all cells have DNA, but not all cells keep their DNA, okay? So sometimes they start with DNA, but they get rid of it. And a great example of that is a red blood cell. So a red blood cell starts when it's young, it has a nucleus, it has DNA, and it makes a whole bunch of proteins. And that protein specifically is called hemoglobin, and that hemoglobin's job is to carry oxygen to our cells. Okay, well, that as that red blood cell gets full of hemoglobin, there's no room for the DNA in the nucleus, so they kick it out. And so then it doesn't have it the rest of its life. So where do we usually find the DNA? Well, you've heard me mention the nucleus. If you're a eukaryotic cell, it's the nucleus. But if you're not the eukaryotic cell, we'll talk about that in just a second. Make you wait for it. But in a eukaryotic cell, you'll also find some DNA in the mitochondria, and you'll find some in the chloroplast. So as we zoom in here on the nucleus, those light blue X's, that's DNA. As we zoom in on the mitochondria, that's this circle that you see here. As we zoom in on the chloroplast, that's that squiggly blue line you see there. That's DNA and all that. Now, remember, the mitochondria and the chloroplast were thought to be prokaryotic cells. And prokaryotic cells, they don't keep their DNA in a nucleus because they don't have a nucleus. Instead, they have a nucleoid, which is just a loose bundle of DNA floating around in their cytoplasm. There's no protection from the phospholipid bilayer around it. It's just clumped together in the middle like a rubber band twisted up. But they also have these little things called plasmids. And the plasmids, pieces of DNA that protect the bacteria is what they are. So who discovered DNA? Well, Friedrich Michner first discovered the macromolecule, but James Watson and Francis Crick are the ones that are kind of credited with finding the shape. So here's Friedrich, and he figured out, but nobody really knew what it was, okay, or what it did. But Watson and Crick sort of figured out, they knew that it was responsible for making proteins, but they didn't know the structure. And Watson's here on the left and Crick's on the right, and they figured out the structure together. But they had a lot of help doing this, okay? They didn't make all the discoveries. So really fast, I want to go through the discoveries. So Griffith here, he figured out that that there was some molecule that transferred information genetically. And Oswald figured out that it was DNA, but people didn't believe him. Chargaff figured out how bases match up with each other. Hershey and Chase backed up Avery and figured out a second experiment to show that it was DNA that carried the information. Wilkins took pictures of DNA, took the first picture of DNA, and this is Rosalind Franklin. She took the best picture of DNA, which Watson and Crick stole, and then solved the puzzle. So what puzzle did they solve? Well, they figured out that the shape is actually a double helix, which means that it's like a ladder that's twisted. It's two strands that go around each other, like a spiral staircase. They figured out this helix has two strands of DNA. A lot of people thought it had three, but it only has two. And they figured out that the strands of DNA are made up of nucleotides. So what exactly is a nucleotide? Well, a nucleotide are the building blocks of DNA, and they're made of three pieces. And so the first piece is deoxyribose, which is a sugar. Remember, we see ose, and we know it's a sugar. And that's where the name, the D part of the name comes from, right? Deoxyribose. Phosphate, you've heard of phosphate in the phospholipids and in the ATP. Phosphate is another molecule that bonds to the sugar. And then up here in the right corner here is a nitrogenous base. And nitrogenous base just means that it has nitrogen in it, okay? So how do we put it together? Well, the sugar and the phosphate join together and then attach to another sugar and phosphate and another sugar and phosphate, and they make the outside of the ladder, okay? In the middle, attached to the sugar but pointing in a, like perpendicular, is this nitrogenous base. And there's different kinds of nitrogenous bases, not just one. And the order of these nitrogenous bases is the secret code. So what are the actual bases? Well, there's only four. And this is why people didn't think DNA was the big molecule. Because there's only four, and it's just their order that makes all the difference. The first one's adenine, which we usually just say A. Thymine, T. Guanine, G. And cytosine, C. So when you see those letters, they represent these words, and I want you to know those words. Here's the chemical, what they look like when they're drawn out. So this shows you where the atoms are in the molecules, and these are all just molecules. So there's adenine, bottom left there. But here's how it all looks together. See how they wrap around each other? See that S and P and S and P and S and P in the pink part? That's sugar, phosphate, sugar, phosphate, sugar, phosphate. And attached to the sugars, you see the base. And if you notice that these bases, they match up with certain things. So adenine always matches up with thymine. And if you need to go back or when we show it again, you'll see that. Adenine always bonds with thymine. Guanine always bonds with cytosine. The way I remember this is kind of silly. A is made out of straight lines and T is made out of straight lines, so they bond together. G is curved, C is curved, and they bond together. That's how I remember. So you look at this. This is a flat piece of DNA. You got your phosphate on the left. The purple is the deoxyribose. Then you got your base. It's bonded, the thymine's bonded with adenine, the cytosine's bonded with guanine, adenine with thymine, and then he goes to another sugar and then another phosphate. And that makes that whole ladder, and then it's just twisted when it's put together. So how many nucleotides are there exactly in a DNA strand? Well, there's 3.7 billion nucleotides in a human genome. That means all the genetic material in humans. If you divide that up for 46 strands in a human being, that puts about 40 million nucleotides into each strand of a human cell. Okay? And so here's what the finished product looks like. All those balls are different atoms, but they come together to make this important macromolecule. So what'd you learn today? Well, we learned what DNA was. We learned why it's important. We learned where we find it, what cells have it. We learned what are the parts of it. We learned how it was all put together. So I think we've had a pretty good day's work. I hope that is clear. If it's not and you have questions, please let me know. Otherwise, I hope you have a great day, and peace out, homies. Copperhead snake.